Hello, good afternoon, friends. I am Abhijit Mane, Assistant Professor of Zoology, ASA College, Ramanand Nagar. Today, we are supposed to know the process of DNA replication. As we know that when a cell divides, each daughter cell must receive its full complement of genetic material in the form of chromosome containing a DNA, or what we know as a deoxyribose nucleic acid. A chromosome is made up of two long strands of DNA and several types of proteins. A DNA strand is composed of a long backbone of sugar and a phosphate unit. One of our different nucleotide bases, that is A, T, G or C, hang off each sugar unit. The sequence of the base encodes the genetic information. The three steps in the process of DNA replication are the initiation, elongation, and the termination. So exactly here we should know how is the process of replication. So where during this process we should know how does a DNA molecule split when it needs to be copied. The second question is, what are the outside rails of the DNA molecule made up of? What happens to the nuclear material in the late telophase condition? What happens to the phase of S phase of interphase? So, we will be discussing about the replication basics. Replication depends on the pairing of bases between the two strands of DNA. The base can only bind to the T and a C can only bind to a G. In the DNA double helix, the base of one strand faces across and behind to those of the other strands. Therefore, the base sequence of each strand complements that of the other. The sequence are antiparallel and serves as a template for each other's replication. The two strands are labeled by the location of certain chemical bonds of the DNA backbone. The cells can replicate the leading strand as a single unit, but must replicate the lagging strand in a small piece. So the next step during the DNA replication is the initiation. Replication begins at a location of the double helix known as ORIC, O-R-I-C, to which certain initiator proteins bind and trigger unwinding. Enzymes known as helicase unwind the double helix by breaking the hydrogen bond between complementary base pairs, while other proteins keep the single strand from rejoining the topoisomerase proteins surrounding the unzipping strand and relax the twisting that might damage the unwinding DNA. The cells prepare for the next step, that is elongation step, by creating short sequence of RNA called primers that provide the starting point of elongation. So with which the next step begins is the elongation. So with the primer as a starting protein for the leading strand, a new DNA strand grows on base at the time. The existing strand is a template for the new strand. For example, if the next base of the existing strand is an A, the new strand receives a T. The enzyme DNA polymerase controls elongation, which can occur only in the leading direction. The lagging strand unwinds in small sections that DNA polymerase replicates in the leading direction. The resulting small Okazaki fragment can contain 1000 to 2000 bases in bacteria. But eukaryotes, organisms having cells with nuclei, have fragments of only 100 to 200 bases. The fragments terminate in an RNA primer that is subsequently removed so that enzymes can stitch the fragments into elongating phase. Finally, the third step starts is the termination. So after a long completion, after the elongation is complete, two new double helices have replaced the original helices. During termination, the last primer sequence must be removed from the end of the lagging strand. The last portion of the lagging strand is the telomere section, containing a repeating non-coding sequence of bases. Enzymes snip off the telomere at the end of each replica, leading to shorter strand after each cycle. Finally, an enzyme called nucleases proofread the new double helix structure and remove mispaired bases. DNA polymerase then fills the gap and create by the exist bases. So this is how the DNA replication is carried off in a cell. Thank you.